because you set it up. So
yeah, and it is
yep,
recorded.
it is.
So everybody be aware that we are now recording. You're on record, official record.
So you have to ask people who don't agree to not talk or log off
you
or
could
something
just
like
say, hi, it's me, but I don't want to talk. That's perfectly fine. Anna, do you want to do a brief introduction again? You're first on my screen.
Sure. I'm Anna Marimashi. I'm working at Duke University, and I've been involved in ontology development. And the reason why I'm here, I wasn't at your session at the meeting because I was attending another session. But the reason why I'm interested to this is because I'm trying to use ontology to help automatic segmentation in images. And in particular, we are looking at the tumor microenvironment. And so that's why I am interested in the representation of the organs and using ontology, integrated different ontology. And that's why I would like to know how are the direction to be, kind of be in the same pathway and not making something that is completely, far from all the other stuff.
Very good, thank you. And then I have David and Jun Lin and then Zorina.
Yeah, I'm David Asumi Sutherland. I'm an ontologist working at the EBI. I'm involved in various atlasing things for the fly brain, for example, fly cell atlas, as well as connected into human cell atlas. And I do some work on the cell ontology and Uberon.
Jun Lee, sorry.
Yeah, I'm from University of Michigan in Ann Arbor. Have general interest in single cell computational analytics and single cell spatial techniques. More in terms of tissue specific atlases, I'm working with the seed network on the female reproductive system. So we would be interested in the spatial common framework for the ovary follicle and for fallopian tube uterus, and on the male side on spermatogenesis. I missed both of the breakouts, so I appreciate some catch up or refresher.
Okay. Zorina?
Hi, I'm Zorina Gallus. As you can see, I represent HubMap. I'm also rooting for New York that I think it's the top of their battle with COVID. I'm not from New York, but we are obviously always rooting for everyone. I'm so called part of the family, I guess, because Kati is one of our mapping centers at HubMap. Obviously, we have an interest, like she said, in bringing organs together and making sense of everything. So I guess I'll stop there. We have a huge storm here. I just saw something flying off the balcony. I hope it didn't hit anybody. So maybe I'll go check quickly. Thank you.
Yeah, now Bruce, then Richard Baldock, then Albert Berger.
I am Bruce Herr. I'm the Senior System Architect Project Manager at Kati's Center. I'm helping to design the CCF ontology with Ellen and Kati and the group. So my main interest obviously is expanding and figuring out the CCF ontology. I have no medical background, so I'm no help there, but will help whenever I can. So.
Hi, I'm Richard Baldock. I work in Edinburgh. I've worked for a long time for the Medical Research Council and the University of Edinburgh, and we developed the Edinburgh Mouse Atlas and related gene expression databases. More recently, I'm now working with Mark Ahrens and Albert on the Gut Cell Atlas funded by Helmsley. And my interest is in the atlasing side and the imaging.
Very good, perfect. Albert?
Hi, this is Albert Berger from Heriot-Watt University in Edinburgh. And as Richard just said, I'm also part of the Helmsley Trust-funded Gut Cell Atlas project.
Andreas, then Griffin Weber.
Hi, everybody. I'm Andreas. I'm a PhD student of Kati's. I work on the registration user interface, which you see behind me. And Serena, we actually had a tree land on our roof last night. So I spent the morning removing tree branches. So I feel you. I hope your house is okay.
So we are getting your storm. We are getting your storm, I guess.
Mm-hmm.
I hope your tree is not coming flying on my balcony.
So Griffin Weber and Amy Bernard, Chris Mungle.
Hi, I'm Griffin Weber. I'm a faculty member at Harvard Medical School and Beth Israel Deaconess Medical Center in Boston. I do medical informatics research. I'm usually working with clinical data, but I did take anatomy when I was in medical school many, many years ago, and I've been pulling out my old textbooks as I work with Kadi on going through Uberon ontology, and in particular looking at concepts that are used to represent the vascular system.
Hi, I'm Amy Bernard. I'm a scientist at the Allen Institute, and our main focus in terms of why I've been involved here is just to try to make sure that we are at least aware and integrated to the extent possible of what's going on with respect to the brain and HubMap. So there's a huge group of people, of which the Allen Institute is part of a consortium that's working on brain anatomy, brain cell types, integrating all of this information from a data-driven perspective into a common framework that includes ontology. And a number of the different people here actually already at this meeting are involved in that through the Brain Cell Data Center. And I'm essentially just trying to make sure that the work that we're doing at the Allen Institute with relation to human brain comes together and sort of integrates as much as possible where appropriate to the rest of the initiatives that are working to integrate the whole human data sets.
Thank you, Amy, for being with us. And we have been able to leapfrog because of all the efforts that have been done for brain. So wonderful to have you here. Chris?
Hi, Chris Mungle, Lawrence Berkeley National Laboratory. I'm here because I work on a number of different ontologies, most pertinent being the Uberon gross multi-species anatomy ontology. And I also do some work on the cell type ontology as well with David.
Ellen, and then Todd Valerius.
Hi, I'm Ellen Cordokas, and I'm working on Kadi's team. I am a biologist, a molecular biologist by training, and I've taught human anatomy and physiology and human genetics and have a good background in all of that. And I've been working on the ontology development with Bruce, and also just generally being a consultant for the molecular technologies that have been coming out. Thank you.
Hi, I'm Todd Valerius. I'm a developmental biologist. I work on the GoodMap project and also the Rebuilding the Kidney RBK project. I serve as a bio-curator for those projects. The GoodMap project is one that's been carried on for a long time, and there's a lot of incredible seed work on ontologies that actually Richard has carried on for many years. And we're trying to build on that into the rest of the lower urinary tract and extend in humans. And I'm desperately anxious, like Amy is, to make sure that the methods we use can link our data out into other forms.
Tim Tickle and then Jim Gee.
Hey everybody, Tim Tickle from the Broad Institute. I'm in the Data Sciences platform in the Klarman Cell Observatory here. I am on a couple data centers, the HCA data center, the brain, the BCDC that was mentioned, and then also the LungMap2 data center. So I'm really just here to lurk and learn, but if I can be helpful, I will try to be.
Thank you. Next one is Jim Gee, and then Aaron Horning.
Sorry, is it me, Kadi?
Yeah, please.
Okay, sorry. So this is Jim again. Here, I guess officially with my hat on for the BICCN Brain Cell Data Center that Amy mentioned. Part of the CCF framework portion of the BCDC.
Hi everyone, my name is Aaron Horning. I'm a postdoc and also project manager at Mike Snyder's lab, and I also work with Christina Curtis. I'm with the ASCT10 and HubMap projects, and I'm really interested in developing the CCF for the colon as it relates to my work on hereditary colon cancer. And so understanding the spatial dynamics of the growth of these things can be really important. And so putting that into a common coordinate framework is really interesting to me. So that's why I'm here. Yeah.
Thank you, Aaron. And I hope you don't have a colon behind you, right? This is just
No, this is a sourdough loaf of bread that I made recently. It's Passover right now, so I can't eat bread, but I can have it behind me, and that's fine.
Okay, good. Yeah, I have seen some amazing photos otherwise from your team. All right, so Carly and then Jenny, please. Carly, you're muted.
Sorry, me, right?
Yeah, please.
Okay, I'm Kristen Ardley, hence the
Oh,
Cardley.
sorry.
That's okay. I am at the Broad Institute along with Tim. I've had a long history of involvement in the GTEx project and the attempts to standardize tissue sampling there across tissues within the human body. So a deep interest there and in how the collections of tissues impact that sampling and the processing as well. Now I'm working on several projects now, including a CZI seed network project and a couple of others that are looking at more detail within tissues. So very interested in this project, and at the meeting we had, I jumped between several groups that included clinical metadata, sampling, and this one. And I think a lot of it comes together in this one.
Thank you. Jenny?
I am Jenny from the Cherry Lab at Stanford, and I previously worked on the HCA-DCP on the data operations team, but now we're part of a newly formed group called the Lattice, and we've been tasked to coordinate data for the seed networks. So I'm pretty much interested in seeing what metadata and data needs to be coordinated for the CCF and going from there.
Very good. And I think we will focus today on the spatial data and on the ontological data, not so much on the clinical data, but I would be happy to also set up one-on-ones, Jenny. Leonard and then Lucy Sue.
Hi, I'm Leonard Cross. I work with Kadi, Bruce, Ellen, and Andreas. I'm part of the MCIU team. I am a senior interactive designer and work on the exploration user interface and the registration user interface, as well as some other projects. And I'm just here to be a fly on the wall.
Lucy, please go ahead.
Yes, this is Lucy Su, and I'm from National Heartland Blood Institute, and I'm in the NIH Hive working group as well. And why I'm here, I think I'm interested in the CCF as well.
Very good. Thank you. And then Lisa, and then Richard.
So I'm Liesl Record, and I'm also with the MCIU group as a part of HubMap. Here, really, like Leonard said, as a fly on the wall.
Hi, this is Richard Conroy from NIH. I'm the program leader for HubMap and involved in a couple of other Common Fund programs, and I'm just here as a fly on the wall as well.
Good to have you. Julie Kim and Chris Briggs.
Hi, this is Julie. I am a scientific program analyst for the NIH. I work closely with Ajay, Richard, Serena as well. I'm here to learn and figure out where I can help.
This is Chris Briggs. I'm with Nils Gellenborg at Harvard Medical School, where I'm senior data curator for the HubMap initiative. So I'm interested, of course, in every aspect of getting data into the system and curating it.
Glad you could join, Chris. So I heard that at least one person would like a quick walkthrough, and I can do this in 5 minutes. So for those which were able to attend our session, this might be already known, but hopefully serves as a refresher of what we would like to do. Let me share my screen here. And can you see my PowerPoint presentation here? So again, can you see it?
Yes.
Yes. Okay, wonderful. So again, this was a session coordinated and organized jointly with Jim G, who is here on the call, and Peter Hunter, who might join us. It was on the Monday, and there were many parallel sessions. We had identified 3 different subtopics. Peter was leading the session on computational physiology. And again, that's another interesting complementary approach to physiology inside of organs that you might like to also check out. It's not the topic of today's call. In HubMap, as you know, we have currently about 9 organs and 25 different assay types, and we are interested in using that data to create an atlas. So we need ultimately data in a more standardized way across the organs, across the laboratories, across individuals, to ultimately create as what I call a reference map over which we then can lay other types of data, including cell types, which many of us are interested in. In our world, we have a multi-level approach where we go from the body to the organ, to functional tissue units, to the cellular level. In our world currently, we have a way to register tissue blocks into reference organs. We get information on the set stacks for these blocks from the TMCs, our tissue mapping centers, and other data producing centers. And then given cell segmentation algorithms or manual cell segmentation, we can then identify the XYZ location for every single cell inside of a 2D tissue or a 3D block or inside of a reference organ. And then of course the reference organ is located inside of a human body. So ideally, we would have reference organs across many of the organs we all care about. We would use collision detection to ultimately register these cuboids and get automatic annotations, semantic annotations using agreed upon ontology terms, and then could run all kinds of interesting queries that are spatial and semantic queries. And so in our setup, we have a so-called semantic annotation workflow. Here in the top left, we have a spatial annotation workflow where we use the visible human data right now to create different reference organs. Yesterday in the TMC call, we got to see a first consensus organ for the kidney. So that's based on 50 different patients. And that gets us more to capture human diversity. Right now, we really literally have 1 visible human and 1 visible female, male and female. And then on the other side, here on the right-hand side, are the interfaces that were already mentioned. So we have the registration user interface that takes a tissue cuboid and helps those which do the tissue extraction register it into a 3D reference organ. And then via object collision, different types of semantic annotation can automatically happen. But those which register tissue can also add their own terms, which then can be checked against our ontology. That all gets stored in our data store, which also has data coming from the TMCs. And then in our current exploration user interface, and also soon via the HubMap portal, you can then run semantic queries and spatial queries. Like you could say, all right, which type of cell is typically next to this particular cell which I'm pointing at or which I zoomed into. So this maybe is more for the second data portal release, but this is where we want to go. And then in our world, we start with a so-called anatomical structure cell types table, and I have a larger version of this on the next slide. And from there, we constructed autonomy. So Ellen, who is kindly taking meeting notes, she is working on that part of the workflow. And then Leonard, who is also on the call, is working with NIAID at the NIH to create these 3D reference organs, which you can then use to do collision detection, but also registration of new tissue cuboids. Just for those which have not seen one of those tables, here is a table that was constructed by the KPMP project. This table, I learned, is also called Pathology and Cell Ontology Table in the UK, if you join from the UK. In here, you have different gross anatomical structures. You have also cell types, and you have marker genes. Now, I already mentioned that within HubMap, we will have many different assay types. So for every single assay type, you might like to add 1 more column here. And so if you are working on different types of markers and assays, this table is also for you. It's just we don't have these additional columns yet. It turns out that these tables can also be constructed for other organs. So thanks to Marta Jorgensen at the University of Florida, we now have a table for the spleen. And we hope that with this call, we could start an effort to get these kind of tables also created for other organs. And specifically what then happens is that these tables are used in our data structures, which then create interlinkages between those ontology terms. So Chris, the ontology terms are right here. And then, however, there are also spatial entities that link to object references. And all these objects have a spatial placement ultimately and get interlinked not only spatially, but also semantically. So in our world, with those two interfaces I already mentioned, we have managed to register tissue cuboids for the kidney. So you see here the left and the right kidney, and also to the spleen. And in the spleen, you have different extraction sites, just like for the colon, and those can be identified. And then all the tissue cuboids, of course, need to end up in those extraction sites because that's where they were extracted from. For the kidney, that's more free form right now, so they can extract from any place in the kidney. Then for the efforts that started last week, Monday, we created a table that lists many different organs and many different efforts. And then during the event last Monday, this table got extended. And so if you go to this web link, which I can paste again to everyone in the chat window, if you go there, you will see a much more expanded version of this table. And you see specifically that also now some other efforts got added and the organs they care about. I also took the liberty to start summing up the totals for per organ and also per effort. And if we at some point get there, I would have more information on actually cell types and layers and data formats for that. But in the interest of time, let's just have me stop here, and maybe Jim Lee wants to add to this. Again, this is not my effort. This is really a collaborative effort in trying to see what synergies exist across the different collaborations and consortia.
Okay, I think that was fantastic. I don't have much more to add. There's a lot of people here. I'd love to hear what they have to say.
So feel free to ask questions.
Can you quickly explain collision? What is that concept?
Mm-hmm. You might have done computer games where you might bump into a wall, or maybe weapon instrument hits another human body, hopefully not. In that moment, two objects collide, and this event can actually be recorded. And if you have two objects intersect each other, that can also be recorded. And so this is a normal functionality that exists in game engines like Unity, for instance, which was actually used for the initial registration user interface. We also have a demo of this, which we gave in a recent tech talk. So Andreas, who I think is on the call, could also just point you to the MP4.
Okay, do you want me to share my screen real quick, or should I just send it?
Yeah, sure. If you have it right there. I mean, it's actually kind of cool that this can be used for semi-automatic generation of semantic terms. And as reference organs become more detailed, then basically the re-registration can be done automatically.
So what you are seeing here, you should be seeing two kidneys now with a cube floating around.
Yeah, I see it.
Okay, so what you see here is a video recording of a, excuse me. What you see here is what you saw there was a video recording of a kidney with the cube floating through, and whenever the cube hits the kidney, it registers which part of the kidney it collided with. It's part of the physics engine of Unity, so it makes call to that. And that way, if we have a three-dimensional object that is deeply annotated like that, you can actually isolate every single collision with various parts of the 3D model at 60 to 100 frames a second. And so this is functionality that we borrow from video games, but which can come in very handy in this kind of automated annotation. Does that answer your question, John Lee?
I think so. I think you are doing automated bumping into different parts in the reference to see if your feature should be assigned there or not. Right?
Yeah, so typically you put one block in a certain area and then all the other 3D objects it collides with are read out and recorded.
Great, okay, thank you.
I have to add that what you saw there was a very, very rough prototype. This is just to see what can be done with 10 minutes of work. If we put several weeks into this, it's going to look a lot more refined. And of course, it depends on the fidelity and the annotation that is already inside the 3D model. But that's something that our team is working on as well, together with NIAD, as Kadi said earlier. Thank you.
I have another question. I think that I've seen that other ASCT table now probably at least 5 times, and I think it finally clicked what it means. And so I think that it would be really helpful to also include that on the ASCT10x10 table, like maybe as a separate tab, so that for all of these groups, we could start thinking about all of these things on what to add for each of these different organs. Yeah, it finally clicked. Thank you.
This is a great suggestion. And actually, we have been discussing how to best make existing tables. So we now have one for the kidney and one for the spleen. And we would love to share that more broadly because right now they have been only shared within HubMap. Well, one is published, so anyone can see it. But the spleen currently is just internal to HubMap. And then going forward, hoping today's call goes well, we would actually identify 5 organs together, all of us, in the next half an hour. And we would have names next to each one of them. And actually yesterday we had a TMC meeting, our regular meeting. And so some organ experts have already said, yep, I can do this, I can take the lead here. So in column T, you have Sanjay Jain listed for the kidney. He actually created this table 5, which inspired the ASCT tables in general. And then Mauda Jurgensen from UFL, she has created the spleen table, and she's now also working on a similar table for lymph nodes while her lab is closed. And actually COVID in this case might help rather than hurt, because people might have time to create these tables while the labs are closed. And maybe these tables already exist in your respective environments. So I know that the Allen Brain Institute, of course, has a table for the brain. It's in a different format, but hopefully somebody is available to reformat it so that it kind of confirms with what we have. And they also, of course, have a reference organ.
Chime in on that David Osumi Sutherland has pointed out that it is there now as of pretty recently. There's a structure that's now in GitHub, which might align to that. So better than what I was trying to pull together before of the soft hierarchical tree. So he's already pulled that in.
But yeah, so that goes back some way actually. So we have mappings from all the brain regions in the Allen Brain Atlas onto Uberon already.
Wow.
That's, yeah, I have a little GitHub repository where I've kind of made all that a bit more explicit. That's actually was Chris Mungall's work about 3 or 4 years ago. That's now being reviewed internally at the Allen. That's the plan anyway. I'm busy trying to turn it into something more table-like to make it more easily reviewable. But yeah, that's
I think one of the one of the tipping points, Kadi, we talked about this before, but with that, it's that we have multiple versions now. So, and the priority, at least for mapping to CCF, had been on mouse, not human. And the human mapping that we have now has been updated with new mapping that Songlin Ding has done. And that is slower to get into the BCDC data just because of the priorities of data generation and mapping that's been happening internally. So while there is something there now in Uberon, the sort of cleanup that David mentioned, my understanding is that that has not happened yet, and it's not yet got any mapping to any of the cell classes. So it's got the structural component, that's a 2D planar structural component for a previous atlas, and now the updated information that Songlin worked on is what would need to be mapped into that new set. So so while there's something there, it doesn't reflect what the data that's actually being generated for all the single cell data generation for human is actually in progress.
So Amy, those mappings are to the ABA brain regions, yes? The cell
Yeah,
type.
so there are two, but that's the thing. There's an old mapping that's not being used for current data collection. So the structures, the anatomy, the ontology for that should it's great. It's a working framework that we had from 2016. The work that's being done now is mapping data into that volumetric reference set, which has some subtle differences and different levels of resolution.
So
it's
maybe we should chat offline to make sure that we're all working to the same versions, and that's always been my fear with this. That so I've got mappings to 2 different human ontologies from the ABA,
yes.
and I just want to make sure it's all current. Okay.
Anyway, sorry to interrupt.
Yeah, and I would assume that we use a staged process. So for instance, stage 1 could be what exists across consortia, and we could just look at this together and find a way to share that with each other in a meaningful way. So for instance, you could have a link to the new Allen Brain Atlas data, the human one, and put all the caveats down that might exist. And then those which work on the human brain could try to resolve those. And then I think what I would love to see is that we meet every 2 months. So in 2 months, we would basically look at what now exists for which organ. And then based on this, we might need to adjust across organs. And then based on this, we would potentially also find a way to detail down to the single cell level. And I know the Swiss team at the University of Lausanne, they have the the Blue Brain browser, and they have an older version of the Allen Cell Atlas or the Allen Atlas for the mouse. So it's not even human, but what they have is a very genius way to have many contribute information on what kind of cell numbers or densities exist for the different brain regions. And I think something like this would be very valuable also for all anatomical structures across all organs. So on my end, on the Hive end inside of HubMap, I will be looking for ways to support, to make it easy for many domain experts to provide that kind of information. But this also requires that we have a browser for different organs where people can select an anatomical structure and then say, all right, for this anatomical structure, I know how many cells of that type are typically inside of there, or what's the density of these cell types is inside of that anatomical structure. And right now we don't have that except for the mouse brain. I'm not aware of any other interface that would allow users to upload that so quickly. So let's see if there are other questions so far.
Hi, this is Albert. Can I just ask David, because he mentioned previously that the other group had some plans or some ideas about how to take this forward. And I'm trying to figure out how these 2 efforts are going to articulate what the idea is.
So I think, I mean, these ASCT tables are fantastic, and they're actually similar things have been worked on in other groups here as well. So maybe I can reach out to the people who've developed similar things, for example, at the Sanger, and we could try and maybe it's worth trying to coordinate there. So there's lots of nice curation that can go on here to map to the cell ontology. I think this is the starting point that I was kind of hoping for these organ-specific groups, and now we can get together. We can see what maps nicely, what's missing. We can get feedback on what's in the ontologies to improve them. So I think if we can maybe center around these tables to coordinate the efforts, that would work really nicely. And so if they can all be hosted somewhere, and if they're updated, then we have some kind of versioning or way to know we're pointing at the latest version. That would be fantastically useful. The one thing that gives me a bit of pause is that clearly, there's already a partonomy in something like Uberon or the various source ontologies, and here we're building another partonomy. So if those 2 things clash with each other, we should at least have some way to validate one against the other and know if there are differences that need to be accounted for. But I think I can work with Chris to at least generate slices of Uberon which give the partonomy just for the terms that are used. So we've been working with filters already to try and do this. So basically we can take a set of terms that you've mapped to in the ccf.al and use that to generate a slice of Uberon with a partonomy, a complete partonomy based on the the semantics in the Uberon ontology, and we can compare the two. So I think that would be a good way forward as well.
And I know there have been efforts to take out all the human elements and put it in a special place, which is still near and dear to Uberon's heart. And I'm so glad we have Chris Mongol right with us here. And potentially, it could also be that these ASCT tables, that they also become kind of a special sub-collection within Uberon. I really don't think that we should start a new set of ontologies.
Yeah, we could use them
try
to
to figure
tag.
out how to have a different view of these ontologies instead, like database views. That would be my ideal situation.
Yep, completely agree.
Chris, would you like to add to this?
I was just going to say real quick, sorry. In support of that, one thing we found in meetings with KPMP and GoodMap and RBK is that if you want more feedback from domain experts that are not accustomed to looking at ontologies, being able to subset those easily makes it a much more manageable workload. So those types of tools are extremely important, I think, to make the information flow into the ontology.
Yeah, I agree. And we've for the FlyCell Atlas, we've been generating reporting spreadsheets to send out for feedback as well. So
yeah, and we can certainly incorporate different partonomy views so long as they're consistent with the overall view. I assume one of the requirements is maybe people like to see a kind of a tree-like partonomy, whereas at the moment, there's multiple different axes of partonomic classification in Uberon, which can be kind of confusing if you're expecting a tree. Is that something that we think we need here?
So Chris, I believe what we can do is to set up an environment, and I envision something that's very similar to the Stanford Encyclopedia of Philosophy of all places. But anyway, that's an environment that's almost like a Wikipedia, but you have different pages, which could just be Google Docs or Google Excel sheets. But I think just a Google Doc with a table in it is maybe the easiest for biologists to operate on. And then you would have one or two people having editorial rights on those pages. And then there is information on how to contact these 2 or 3 experts so that you can have your voice heard and changes made. And so initially, we would set this up for the 5 organs we are on the hook for HubMap for next year. And then, of course, we could easily make another page for brain, which we would love to have. I think it's just sad to look at a human body without a brain. And it's just copying over the fields, right, the standard form over and assigning 2 or 3 editors to it. So I think that's what I can promise with Richard Conroy watching me here. But I think in the interest of us all learning from each other, it would be beneficial to actually see how these pages get filled out, what level of detail we have, what caveats still exist. So the editors could then have all kinds of disclaimers added to those websites also. And again, for us inside of HubMap, we need to have these pages anyway for at least 5 organs. And there might be others which would like to come on board and work with us on this. And again, ultimately, this is a humongous effort. It takes much time to walk over these terms over to Uberon and make sure that these names are all correct. It's a lot of work to do those 3D reference organs, but hopefully we can all benefit from each other and do this together, as opposed to each one of us does something and it doesn't even fit at the end.
Okay. Yeah, I have some questions about how that might exactly work, but that's maybe a topic for a future call.
Yeah, I think we need to be careful about sort of the balance of the sort of informal wikiized way of working and how that gets incorporated into the formal side. Because if those 2 things drift apart, you just end up with something that nobody can use. But I completely agree. We need quick ways to generate slices and views with names that work for people and that have only the human terms, for example, and then quick ways to get feedback back in. And I think we're quite close to being able to do that. So,
and actually, within HubMap, we originally had this kind of information collected via GitHub, and just accessing GitHub we didn't get it to work. I mean, even with a lot of hand-holding, it wasn't possible. So yeah, it would be great to actually automatically read out those tables and just put them into a mechanism which then automatically tries to walk them over to Uberon, and then what cannot be matched automatically has to be done by hand. So I think automatizing this entire process is an act. On the other hand, I think there are about, I don't know, 1,000 anatomical structures that we initially might be
It
caring
can be
about.
curated. It can be curated, especially if it's split between different groups.
Yeah, yeah.
I think.
So right now, I would like you to focus attention on columns S and T in that big table we have in front of us. And every one of you, and the entire world actually, currently has editing rights on this table. I do save it from time to time because I don't know what's happening next to this table, and it seems valuable to many. But right now, any one of you could actually start voting for certain organs. So if you have a favorite organ that you really would like to have included into an initial set of organs that we could all collaborate on, I would like you to add an X. So for instance, in the S4 column or cell, you have that we have a draft. So HubMap has a draft. We are very happy to share this. Similarly for spleen, whoever would like to see it, please just let me know. Would like to view, maybe I make another column in V, would like to view. And then ultimately, I would love to see organ expert 1 and 2 in the T and U column. And I know this seems like a humongous commitment right now, but maybe you're also excited and okay to put your name down there with the understanding that life happens. And right now, much life happens because of COVID-19. So we won't hold you, you can always tell I can't do it anymore. But I would love to see if there are volunteers which would love to work on certain organs. And Todd, thanks for putting your name down on the kidney. And based on what expertise—
Who did that? Hold on.
Oh, I didn't do it. I'm not doing anything anymore. Oh, all right. I got a shock for a moment. I don't want you to not want it. So yeah, if there are people here on the call, or if you know of people that might be interested to serve as organ experts, which really many of us on the call here are not, that would be very important because we need this anatomical, pathology, medical expertise to get us these initial table. And again, initially they will not be complete. They might not even be correct, but it's a matter of getting a first version so that we can start discussing it.
This will be information, on the structure, cell, and markers you're talking mostly, or even the spatial relation between the different.
So right now we need tables like, let me bring that up on my screen again. So we are talking about tables like this one, which we got for the spleen now, or like this one, which we have for the kidney. And as you see for the spleen, there is a marker subset. But again, there could be other columns extending to the right, which might have other types of ways to identify cell types. And if you cannot provide marker subsets, that's okay. I think if we just have the major structures and substructures and ideally also cell types associated with those structures, that would be wonderful.
Okay, and this is human only for now.
Yeah, right now we would be focusing on human.
And
just another point I wanted to highlight with brain that's going to make this challenging is I know that for some of these we're looking at cell types that are well known because they're associated with clinical identity or with well characterized functional behavioral properties that are well understood in some of the other organ systems, particularly metabolic organ systems. One of the challenges with brain is that once you get down past a very, very high level of things like neurons and non-neurons or excitatory and inhibitory cells, there's very poor alignment on what those cell type classes are. So that's a big problem right now in brain. It's a very sociological problem. It's like a problem that's been around for 100 years of what those types are, which is why I before indicated that for brain, we have some of the anatomy, but mapping to those types is going to be a really huge mess. So one of the ways the field is aligning around this is to specifically look to the single cell transcriptomic data to inform that. And why that's sort of a chicken and an egg problem is because we don't have that data for whole human brain. So that's where, for what we do have right now is for the motor area of cortex. That's the information that I shared with you, Kadi, related to just what we've been able to sort of loosely map at a general level. I've shared that now also with David and Chris, just in a separate email. But we can't do that for the whole human brain. We can just sort of mock it up as there's going to be structures and there's going to be some sort of high-level cell classes, but we don't have the information. And so even though sort of organizationally in terms of CCF world, we've got a lot of progress in brain, at the level that maps down to this kind of specificity, it's going to get messy very quickly. And this is essentially what the entire brain initiative is about. So the BICCN project that includes the data standards and data sharing that the BCDC team is doing, that James and David and Tim and others here are all, you know, involved in regularly. It's all to try to come back around to that. But I just wanted to give that perspective for the brain, because that's why it's not a complete set right now.
Yeah, no, I realize. And again, it will not be complete and it might even be imperfect initially. But as long as we agree on this, on a certain workflow and in a certain process and how we even capture that data, then I think also other organ experts can join over time. And it's good to start with a small subset, not with all 37 organs, which we have listed here, because that's just a massive undertaking. So right now I see kidney, and let me just share my screen so that we also have it all recorded here. So in this part, I see we have kidney captured right now, we have spleen, we have the large intestine and the small intestines. Thank you so much for volunteering there. Then we would have the lymph nodes, and then we have prostate. So I think that's a wonderful set to start. I don't see brain. I hope there will be some brain volunteers in here.
See, I'm worried already because I see brain and cerebellum are separate. That's horrifying.
Well, somebody asked us to do that. I didn't do that out of my own devices. That's
anyway, I don't want to get into the specific structures for brain. I think it's premature for, I mean, I think given that there's a whole initiative working on brain, you know, it'll get covered. But in terms of its completeness criteria, I think it's going to be early. I think the team's working on it. Just my 2 cents.
Maybe we can unify them by color or something, yes.
So this is Albert. I'm working on the gut salad list. There's obvious interest in anything related to the gut, but I'm not the domain expert, so I'm quite reluctant to put my name in there, but I'm equally reluctant to put the domain expert's name in there without having talked to him first. So I think this is maybe just a touch premature to ask for names on there. Certainly, I'm not sure Richard will agree that we have a very strong interest in doing that for the gut and participating or leading, whatever, collaborating with people. I'm just a bit wary of putting names there without having talked to the domain experts.
Yeah, and Albert, feel free to talk with them. And if we see a new name in the next 10 days, we can still involve them and send them the information that everybody else gets. So feel free to please do talk with them. They should feel like they really want to do this. The other way of going is also that if you know of other efforts that have worked on these kind of anatomical structures, cell types, tables, if you want to just point me to relevant literature or relevant experts which might be interested to collaborate there. So again, this will be a phased approach. Initially just 5 to 10 organs, maybe just 5 to really get those processes down. And then again, in 2 months, we would meet again and see if we now have more clarity on those 5 organs. And then maybe others want to join because the process seems to work. And maybe we can't get it to go, but I hope we can, because I think the organ experts, they seem to be able to provide these tables. And again, on our end, this is gold, because then we can design the ontologies that are needed to support this. We can use those terms to label the 3D reference organs. And again, inside of HubMap, we have quite a number of organs for which we need to do that. Are there any other comments, questions on process? Are there any other pieces of information that you would like to have? So I think what will happen is that you all get an email with a thank you for attending today. If nobody argues against this, I would like to share this recording with everybody who wanted to originally attend. I know Sarah Teichmann has been trying to make it work, but she couldn't attend this very time. Peter Hunter also wanted to attend. I think he must be busy with the PPE production they are doing for New Zealand. So these are all good reasons for not attending. So if you don't mind, I would love to share this recording. Is anyone worried about sharing this recording?
No.
Thank you. And then I will call another meeting in 2 months so that we see what progress was made. But I will also start together with Ellen on our team to start interlinking with the organ experts we now have listed here, or we will have listed in 10 days, and see what we can do together. And then we would present back in 2 months what has been accomplished. Todd, I think you wanted to say something.
I just want to ask what pediatric systems meant in the list.
Pediatric. Where do you have it? Can you highlight?
On 20.
20.
Yeah.
Well, that's a good
23,
question.
yeah.
I did not add
Okay.
that in there. So now we could see who is working on it. So it's either H10, and we have Aaron on here. I don't know, he might know. And then there's also Welcome, which works on that.
So I just want to comment that when I was assembling this table, I was going based on websites and for the consortium. And so if that's what they put, that's what I got put in the system. So in the table.
It makes sense. Thank you.
So apologies if I should be more specific on that, but that was kind of the general, we work on pediatric systems. So
I wonder, given, we just had the HCA meeting last week, and there are HCA interest groups for major organs, which, collectives of groups working on that. That seems to be the best place to tap up, or a very good place to tap up for expert input. And I wonder whether in the meeting there was some discussion of kind of organizing jamborees for specific organs, where we have organ-specific meetings with a certain amount of prep, and then some live work on this as a way of kind of galvanizing people to do it, rather than it being piecemeal, a couple of experts looking at stuff. Do you think that could work?
Could you give us a list of the interest groups that exist within HCA? I'm not familiar with it.
I think is Tim Tickle on still?
I'm on. Hey.
Hi, yeah. I'm guessing you might be the best person to be the bridge across to the different HCA groups. So,
as far as like the biological networks.
Yeah.
So Katie, I'd be happy to reach out to you in an email and connect you to the office, and they'll be able. There's one or a couple people that are leads for each biological network. They might be a good place to start.
Yeah, thank you for pointing this out. I think whatever we can do to leverage existing infrastructure, existing expert networks, thank you for these bridges.
Also, I'd like to mention to Amy, the reason cerebellum was also parsed out is because that is the part of the brain that that particular consortium was working on. So HCA listed cerebellum as a specific target, not the entire brain. So that's why that is listed separately in the table.
I think that's great. I think the, I mean, we're sort of conflating two things where there's project teams versus if we're actually looking for the ontological structure set. Yeah. So I think they all need to be addressed and can be addressed, but we shouldn't conflate that sort of first left-hand column with the ontological organist system. Yeah. So if people are looking for, an expert or a go-to person, that's in essentially managing what that anatomy means and where it goes. Yeah. So it's not necessarily one and the same that's going to run along that row that's in the column. But yeah, I just actually added a hyperlink in the tiny little X under brain to David and Chris's Uberon reference for brain, just so people know where that's there. It's really super hidden, but I can't
see it. I'm worried now. can you maybe put it underneath the table, like down here in the?
I could. I just wasn't
52
sure
or
what the best place
53?
is to add it, and it's sort of, it's not owned by me. It's David and Chris that are the experts, and just even what level of confidence they have. I'm just putting it there to aggregate some information.
Yeah, I don't see an annotation here. So if you can just put it underneath the table in the long listing of all kinds of additional information, that would be great. Like line 52, that would be great. So I think we are out of time, everybody. But this was wonderful, and I'm looking forward to stay in contact with email also. And then David, if there are other HCA NIH meeting sessions going forward with concrete action plans, please keep us informed also. Again, I'm in contact with some of them, but might not, I mean, I was definitely not able to attend all of these parallel sessions.
Mm-hmm.
So if you see synergies there, please do let us know.
Yeah, I mean, I think I can try to, but I'm probably not the best person for that. I mean, so I guess Tim is closely connected with a lot of this, and maybe we should try and get some other HCA, DCP people involved for that kind of coordination.
Yeah, I mean, yeah. So there might be. I think that like Laura Clark was also attended, and she's really into this these areas of concern too. So I can CC her as well, Katie, and maybe we can talk through a strategy on how we can do that. But great feedback though, David.
Yeah, wonderful. Thank you all for your time, and thanks so much for volunteering also. And again, if you know of others, we will look at this table in 10 days, and whatever names we see there, make sure we have the email addresses also. We will contact and share more information for this initial phase 1 engagement here.
Nice job, Katie. This has been really great.
Thank you all.
Thank you,
Bye.
Dottie.
Thank you.
Thank you.
Speaking. Thank you.
Bye-bye.